without further ado, we will go to our first speaker, who is somebody that you may be familiar with, Nuno. And Nuno is a postdoctoral fellow here in CCBR. He is in Benjamin Barbosa. He completed his undergrad in physics and engineering at the Technical University of
Lisbon,
in Portugal. He did his PhD in Lisbon and Cambridge,
yes,
a joint
yes,
appointment, and then also postdoced in Cambridge under Simon Tavera. And currently he's the postdoc in Benjamin's lab, and he's going to speak to you about his work there.
Okay, thank you, Michelle, for the introduction. Given that this is a very short talk, I'm going to use it as a teaser for a larger project, okay? But it's especially for the most junior members of the audience. And the idea, I'm not going to go through any relevant bioinformatics tool. I'm actually going to show you an example of how simple bioinformatics tools can actually have quite an impact in addressing what I believe are important and basic biological questions. Okay, so essentially, the Blanco lab is interested in the regulation of alternative splicing and also in the way alternative splicing has evolved. And I will start with a very classical conundrum, which is why, you know, different beasts like these two that at least, they do share some similarities but have some things in different, actually have a very comparable number of protein coding genes. And we believe that part of the answer lies in mRNA splicing. So, you know, actually the concept of gene expression, as you've heard in previous talks, including the one from Mike Bruno sitting here in the first row, is that this concept is a bit obsolete, and one of the reasons is why, you know, a gene product can be many things, right? You can have many isoforms, and this isoform diversity can come, at least partially, from this process or this step in the processing of the RNA, and namely the messenger RNA called splicing, in which some bits of the premature RNA that just got transcribed from the genome get spliced out, we call the introns, and then the remaining bits that are spliced in together we call the exons. And not only this happens, but this can happen in many alternative ways, what we call alternative splicing of the RNA. And the way this is regulated, and that's what the lab is studying, is by an interplay between sequence elements, you can call the cis-acting elements, so motifs, more or less degenerate, more or less specific sequence elements in the RNA that do tell what we call the transacting factors, the splicing factors, protein elements. Basically, they tell the protein elements where to act, where to bind. Okay, so these boxes here are the exons, the bits of the RNA that will survive splicing, and in the case of protein coding, be translated into peptides afterwards. And here you have an example of two exons that are always spliced in, and one alternative exon that can be spliced in or skipped. That's basically what we are showing here. So this exon skipping or cassette exon splicing is the most common type of alternative splicing, and that's the one I'm going to focus on during the next 10 minutes or so. But there are many other, there are basically all the combinations you can think of can happen in terms of transcripts being alternatively spliced. And as I told you, based on the first slide on the conundrum, one of the reasons we believe alternative splicing might explain at least partially the tremendous phenotypic difference between, you know, different species is not only because alternative splicing allows for the generation of an amazing diverse set of isoforms for each gene, but we actually, work done in the lab already suggests that alternative splicing seems to be frequently species specific. So the way alternative splicing is regulated, or the way genes are alternatively spliced, seem to be a species-specific thing. And we made this observation by comparing both human and mouse, and actually human and chimp that are quite close in evolution. The other reason why we think that alternative splicing might provide a good source of phenotypic difference between species is that when you actually quantify the percentage of genes that undergo alternative splicing in the different species, what you see is that there's a sort of correlation with what you would call complexity, right? So in vertebrates, actually most of the multi-exonic genes undergo alternative splicing, and that's not the case for simpler organisms. So for the fans of Ford, it's okay. Ford is more complex than a worm, it's fine. Anyway, this can be even more interesting if we know, and this is work done here in the building, Tim Hughes and Quade Morris labs. They've shown using microarrays a few years ago that when you look at gene expression from different homologous organs across different species of vertebrates, so when you look at gene expression profiles of orthologous genes, these samples do cluster by tissue type, by organ, and not by species. So in terms of gene expression, my liver is more similar to a frog's liver than it is to my brain. With RNA-seq, so the new high-throughput sequencing technologies, Henry Keismer's lab has also demonstrated the same, and here you just have a principal component analysis plots showing that samples in terms, again, gene expression measured this time using RNA-seq, they cluster by tissue type, which are the different colors here. Okay? So gene expression seems to be, or a tissue-specific, tissue-regulated gene expression seems to be conserved across vertebrate species. And so it's not gene expression that is very likely to account for the majority of these phenotypic differences. So what is it then? We think alternative splicing might contribute, so we asked the question, how has alternative splicing evolved in vertebrate species, where it is quite prevalent? And how did we do that? Well, we got RNA-seq from physiologically equivalent organs from this set of vertebrate species. So we got our own data, and also we downloaded published data. So you can do this sort of analysis without spending money on sequencing. This is a very important point, because there's a lot out there already. And amongst the vertebrate species, we have species that, frog, it's more than, it's actually around 380 or some million years apart from all the others. But we got species as close as these two. Okay, so we really had the full resolution. And what did we do? Well, simply we just got polyadenylated mRNA and fragmented and get the cDNA, use Illumina HiSeq, which you heard about, to sequence these approximately 75 mer reads for each molecule. And then we went for a very simple approach to look at alternative splicing. We didn't quantify isoforms. We have an exon-centric approach, and we just look up the splicing of each exon. And so basically we quantify the reads that map to the exon-exon junctions. For each exon, we looked at reads that map to the constitutive junctions, the ones associated with that exon inclusion, and the ones that map to the alternative junction, the junctions corresponding to the skipping of an exon. Okay? And then we did this not only looking at the differences between tissues in the same species, but we look at orthologous exons in between species. So the example here is chicken and human. And we ask ourselves where are the differences, and if the relative frequencies would actually be difference between species. So the first step, we actually just quantify. Before I say that, just a side note to tell that we use liftovers, which is a simple tool that you can find in Galaxy to determine the exon-exon orthology. And first question is, do we have differences in the relative abundance of exon skipping of alternative splicing between species and between organs? And the answer is yes. So what we find, first of all, and this was already known, is that neural tissues and testes have more alternative splicing than the other tissues. What was not known is that it looks like primates. So here you have all the species we've used, and they are mapped according to their evolutionary divergence from human. Primates seem to have more alternative splicing, or at least more exon skipping, than the other vertebrate species. And just for curiosity, the genes with more alternative splicing complexity in human seem to be enriched in cytoskeleton-associated functions. So you can already start speculating in terms of what it means developmentally. So that was just quantifying evidence for alternative splicing. But then we actually look at the actual numbers, and we compare exon by exon across species, the different levels of inclusion. How do we quantify this? Well, what we do is just determine for each exon what's the percentage of isoforms, and we call it PSI, percent splice gain. What's the percentage of isoforms that include the exon? And it's basically just, you know, the average number of these two over the total number of reads that map to the junctions. Just a note to say we correct these read counts for the total number of uniquely mappable positions in the junction database, in case you're interested. And what we find when we take this huge matrix with all the exons and all the inclusion levels across all the samples in all species, when we take this matrix for all the exons that show some evidence in at least one sample for alternative splicing, when you cluster all these samples together, they cluster by species with no exception. Remember that for gene expression we have these things clustered by tissue, for alternative splicing they cluster by species. And essentially what you're seeing here is, if you ask in a Shakespearean way, to be or not to be alternative, what this is reflecting is that for an exon to be or not to be alternative is a species-specific thing. So when you take all the exons that are alternative in human and you ask how many are alternative in the other species, you see this rapid decline. And even chimp is only about half in common. Okay? So alternative splicing apparently a very species-specific thing. We did also gene expression, and we got the same results as our colleagues did before. You know, gene expression totally clustering by tissue type, not by species. There is, however, one interesting exception here, is that if you take, I told you most of the exons are not, I mean, being an alternative exon is not something that is conserved. But there are a few exons that are alternative in many species. So we select 4 species that are lineage representative: human, mouse, chicken, and frog. And this is a Venn diagram. You can't see it properly. But we asked ourselves, which exons are alternative in all these 4 species? And we found 41 of them. And when you look at the inclusion levels of these 41 exons, interestingly, the ones that are conserved as alternative do cluster by tissue type. So the ones that are conserved as alternative seem to be tissue-specific, tissue-regulated. That seems to be conserved. And a different way of illustrating this is if you take all these categories of the Venn diagram and you look at the distribution of inclusion levels, what you see is that exons that are alternative in only one species seem to be almost always included, so closer to the 100%, 90-something%. But as being alternative becomes a conserved thing, what you get is if you have exons that are alternative in many species, they are more regulated. So you see that the inclusion levels spread across the entire scale. Last meaningful slide here before the acknowledgements, just to let you know that, and I'm not going to, this would be another talk or another 10 minutes, but the other interesting aspect of this work was that in collaboration with Brendan Fry's lab, in 2010, the 2 labs published this interesting paper about the splicing code, which basically is this algorithm that uses combinations of hundreds of different RNA features and tries to predict tissue-dependent changes in alternative splicing, tissue-specific alternative splicing. This was done with mouse features, and what we did, and I'm just giving you the example of increased inclusion of an exon in brain, what we realized is that when you apply the mouse code to other species, you see here that you get very similar performance. So it looks like this, although different exons are regulated, the core set of RNA features that I use to regulate alternative splicing seems to be a conserved feature in vertebrates. And as I told you, this was just a teaser. It's a much bigger project, and it was a teaser for you to read our paper. So this has been published recently in Science. As you see here, it was the Christmas issue, the Higgs boson issue. And there was, although I'm the lead author, there's lots and lots of people involved. So apart from a lot of people in the Blanco lab, here at U of T, not only Brendan Fry's lab, but also Phil Kim's lab, Mike at Sick Kids, and collaborators in Cambridge. I'm just acknowledging here the two funding bodies that supported me here during my stay in Toronto, which is about to come to an end. And the final slide is a request from Ben to advertise Bioinformatics postdoc position in the lab, so I can be replaced. Okay? Thank you very much. Questions? Alex?
Is alternative splicing responsible at all for the phenotypic differences between populations
Between?
Populations and humans?
Actually, I don't think that has been studied to a degree. So the only answer I have for you is there are you have association between SNPs and mutations and so on and alternative splicing patterns. So I would expect that even to a small extent you would have that. Yeah. Question there?
Have you considered trans-splicing events?
Not in this study, but we are looking. I mean, yes, those are a bit harder to quantify and to model. So I don't have for this data set an answer on trans-splicing.
Do alternative splicing events generally preserve the coding frame of the protein? I mean, I assume they do.
The ones that you can measure do.
Okay.
So that's the thing. So what happens is that there is a lot of alternative splicing that might be happening and we don't know, and there is evidence. So what happens is if you preserve the coding frame, you will have a protein.
Yeah.
But if you introduce a permature termination codon, the RNA is likely to be degraded by nonsense mediated decay, right? Nonsense mediated decay, which means that you won't be able to measure it with this technology. However, there is already evidence. You have a lot of literature showing that introducing a premature termination codon and inducing nonsense mediated decay is actually a way of regulating gene expression. So instead of playing with transcription, you might decide just to, screw up the message so the mRNA gets degraded instead of still producing the same amounts. Final one?
Yeah. Have you or do you know anybody who has tested if the ultimate proteins are also tissue-specific or species-specific?
At the protein level. Yeah. So one bit of the work that we didn't show, but you can read it on the paper, is that conserved alternative splicing tends to go for high disorder areas of proteins, which are supposed to be associated with, or at least more likely to be associated with protein-protein interaction. So it looks like the conserved, let's say, modeling of the protein structure at the level of alternative splicing might be at the level of, disordered areas of the proteins that are associated with protein-protein interaction. I'm not sure if this totally addresses the question, but we have at least a hint based on that. Okay? Thank you very much.
Thank you.